Validation and verification. This process has become important component of every sector to maintain quality of product and service. In this video, we will talk from clinical laboratory side. We know that clinical labs are the end user of machines and methods. So to provide quality report, lab must align with validation and verification process. As we know, for standard practice, it is better for clinical lab to be certified from ISO, and ISO 15189 is there to look after medical laboratories. I'm connecting this with ISO here because this validation and verification are important component of ISO certification process. See, in ISO 15189 2022, there is detail about validation and verification method in the clause 732 and 733, where after verification and validation, documentation is very important step. Now let's dwell into the topic. To make it easy, I'm starting with who do validation for clinical lab and who is responsible for verification. For validation, manufacturer or test developer is responsible, and in some condition lab also need to do validation. This means when manufacturer develop new machine or test method, they need validation before selling it to the market. On the other hand, the clinical lab needs validation when they use non-standard kit. Let's say kit that is not approved from FDA or CE. At this stage, they need to validate the kit before using them. Another case, the clinical lab needs validation when they modify the manufacturer test method which is out of scope. For example, if they change the sample type from serum to urine, or manipulate the reagent volume or test volume, or something like this needs validation. Also, when lab develop their own test method, they must do validation for further use. So the validator are manufacturer and in some cases clinical lab. Your manufacturer and clinical lab. Now the verification. The verification is conducted by laboratory staff as per manufacturer instruction and ensure that it is okay for routine use. The laboratory staff works as per manufacturer instruction and then they ensure that it is okay for routine use in the sample of lab. Okay, so let's say we purchase already validated test kit from supplier. We calibrate it in our lab, run control, and after QC pass, we verify this is okay for routine use. So the verification is done by lab staff in their own setting, own reagent machines, and in their own environment. Okay, what defines validation and verification? Validation means to ensure product is built correctly and meet all specs and designs. Here it proves that method worked as planned. Now the manufacturer can sell it globally under defined condition. In case of lab, after validation, it can be used for public platform. On the other hand, verification ensures product meets user needs. It proves that the test method works well in our lab setting. Now can go for routine use. Coming to the parameters, there are some major parameters required for validation and verification of test methods. These are the parameters. To validate new method, sensitivity, specificity, linearity, precision, accuracy, stability, reference range. These are some major parameters. There are other also. So they must meet the requirement. And for verification, precision, accuracy, reportable range, reference interval must be addressed. We'll talk about them later. Okay. Now frequency. The validation is done once per method by test developer or manufacturer, or we can say before release of product validation is done, but the verification is regular. Every time we run different level of quality control sample in our lab is a part of verification. So we can say that verification is more frequent in the lab and validation is very less frequent. It is done once by developer or also in lab. It is done once before product release, before launching it. But verification is regular process. Let's see how things go for validation. To develop new test, the developer must satisfy this parameter. Sensitivity. It determines limit of detection, that how small quantity method can measure. Specificity. It checks the interference of hemolysis, lipemia, bilirubin and so on, and also cross reactivity with similar substances like antibody and antigen. For linearity, multiple known concentration from low to high should be run and graph is plotted. And for precision, repeat test across the day and operation to get desired CB. For accuracy, compare your result with reference method and lab. And for recovery, spike the sample and calculate recovery percentage. The ideal recovery percentage is 100%. For stability, check sample and reagent stability at different level of temperature, pH, humidity and others. And reference value, it can be established according to the local population if necessary. Now approach for verification is here. Check precision of method. For this, lab must run control and calculate CB percentage. To check accuracy, compare your lab result with reference lab or participate in external quality assessment program to know your deviation status. For reportable range, test low and high calibrators to ensure the linearity. And for reference value, you can align with manufacturer or establish your own. Here are some examples of validation and verification process in lab. Here you can see developing ELISA test kit is the process of validation. Creatinine test as per instruction. Here you are doing the test as per instruction and this is verification. Changing the sample type. Here sample, you switch sample from one type to another. So you need validation now. This is validation. Example. And doing HIV test from FDA approved kit, then it is verification. Okay. Thank you very much. This much for today. For any query, you can comment in comment box.